Okay, so I apologize in advance. My neighbor's fire alarm has been ringing like non-stop, so please try to ignore that. Sorry for the inconvenience. So this is muscle tissue. We're going to be talking about muscle tissue. Muscles, muscle functions, the three types of muscles and their distinguishing characteristics. Isometric, meaning same measure, versus isotonic, meaning same tone. Aerobic means oxygen present, so aerobic respiration. Glucose plus oxygen gives CO2 plus water plus 36 ATPs. Anaerobic, no oxygen available. Anaerobic respiration, glucose equal pyruvic acid equal lactic acid. Lactic acid causes muscle aches and cramps. Muscle forming cells are called myoblasts. Some muscle proteins are myoglobin. Myoglobin produces the red color of muscles. Actin are thin filaments that slide during contraction. Myosin are thick filaments that are stable during contraction. The neuromuscular junction is the space between a muscle cell and a nerve cell. It is also called a synaptic cleft or region of synapse. The neuron releases a neurotransmitter, a chemical called acetylcholine, into the cleft, and this causes stimulus in the muscle cell. The sliding filament theory explains how actin filaments slide parallel to myosin filaments. Then the heads of myosin contain ATPase, which is an enzyme that catalyzes the splitting of ATP. Calcium is used to bind actin to myosin heads for cross-link action. The cross-link formed is broken when calcium is released from the bond. If calcium is not released, then muscle stiffening occurs, as in rigor mortis. When stiffening occurs 3 to 4 hours after death, peaks occur up to 12 hours, then dissipates in 48 to 60 hours. Hence, coroners can predict the time of death. Characteristic of muscle: excitability, which is the ability to receive and respond to stimulus. Contractibility, which is the ability to shorten when adequate stimulus is received. Extensibility is the ability to stretch or extend. Elasticity is the ability to resume its length after contracting or stretching. So the four characteristics of muscle are excitability, contractibility, extensibility, and elasticity. Some definitions are muscle fatigue, which is a state in which the muscle is unable to contract and its tension drops to zero. Inability to contract occurs after vigorous muscle activity. Atrophy is degeneration and loss of mass due to disuse. Hypertrophy is increase in mass, as in bulging biceps of professional weightlifters. Dystrophy is muscle destruction by disease. So atrophy is degeneration and loss of mass due to disuse. Hypertrophy is increase in mass, like in weightlifters. Dystrophy is muscle destruction by disease. Oxygen debt is the extra amount of O2 that must be taken in by the body to provide for restorative processes. such as the conversion of lactic acid to glucose or glycogen in the liver. Running 100 yards in 2 seconds gives accumulation of lactic acid. Pay back oxygen debt by rapid, deep breathing. So the 3 types of muscle tissue is cardiac muscle, skeletal muscle, and smooth muscle. So this is the cardiac muscle. This is what a skeletal muscle looks like. And this is what a smooth muscle looks like. Cardiac muscle cell, skeletal muscle cell, smooth muscle cell. And these are models of each muscle cell and what they look like. As you can see, the cardiac and skeletal muscle have striations, but the smooth muscle, which is this one, doesn't. 3 types of muscle tissue in death is skeletal muscle. Skeletal muscle is striated. It's voluntary. You have conscious control over it. It's found on the skeleton, and cells are cylindrical and multinucleated. Okay? Cardiac muscle is also striated, involuntary, unconscious control. Only found in the heart. Cells are branched and uninucleated or binucleated. Smooth muscle is non-striated. It's also involuntary, just like the cardiac muscle. Found in walls of hollow internal structures, cells are spindle-shaped and uninucleated. Properties of muscle tissue. Some functions of muscle tissue. They produce body movement. They stabilize body position. They store body substances, such as food in the stomach. Then they generate heat. So muscle tissue produces body movement, stabilizes body position, stores body substances, and generates heat. Some properties of muscle tissue, again, is excitability, contractibility, extensibility, and elasticity. Some smooth muscle functions: forms around other tissues in digestive and urinary systems, forms sphincters, produces contractions called peristalsis. So smooth muscle is found in the digestive and urinary systems. In integumentary system, erecti pili muscles cause goosebumps. In blood vessels, regulates blood pressure and blood flow. In reproductive and glandular systems, smooth muscle produces movements of products. Cardiac muscle is present only in the heart. The cells are striated and branching. This is an example of branching. Ends of cells are joined by communication junctions that allow the cells to contract as a unit. So this is a junction between adjacent cells. Coordination of cardiocytes. So cardiocytes are cells of the heart. So because intercalated discs link heart cells mechanically, chemically, and electrically, the heart functions like a single fused mass of cells. That is amazing. Because intercalated discs Okay, yeah. This is basically the same slide repeating. So, these are some numbers about your heart that may surprise you. Some fun facts. The system of blood vessels, including arteries, veins, and capillaries, is over 60,000 miles long. It is long enough to circle the world twice. The heart beats about 100,000 times a day. In a 70-year lifetime, the average heart beats more than 2.5 billion times. An adult woman's heart weighs about 8 ounces and a man's about 10 ounces. That's amazing. Histology of skeletal muscle. So this is what skeletal muscle looks under the microscope. Specific functions of skeletal muscles. So, skeletal muscle produces skeletal movement, maintains body position, supports soft tissues, guards the body openings, and maintains body temperature. So, what are the 3 microscopic differences between the 3 types of muscle tissue? So this is smooth muscle. These are the nuclei. Okay. This is cardiac muscle. And you can tell this is cardiac muscle because not only do you see the intercalated discs, you see the striations. Okay. And you see branching. These are myocytes, these are nuclei, striations, intercalated discs, and this would be skeletal muscle. The properties of muscle tissue, again, are excitability, contractibility, extensibility, and elasticity. So what is excitability? You can answer that. So this stands for neuromuscular junction. The neuromuscular junction, this is the nerve-muscle relationship. So these are models. The neuromuscular junction. This is the sarcolemma. This is the synaptic bulb. This is the motor neuron axon. And this is the endomysium. So sarcolemma, synaptic bulb, motor neuron axon, endomysium. So this is the superior view of the neuromuscular junction. You can see the sarcolemma, the myofibrils, which is in this circle. These sections are myofibrils. Okay, sarcolemma, this is the sarcolemma, the sarcoplasm, the nucleus, the axon, synaptic bulb, okay, synaptic bulb, myofibrils, it's the interior view, the axon, myelinated, endomysium, synaptic cleft, sarcolemma. So the neurotransmitter acetylcholine, or ACH, travels across the synaptic cleft, binds to membrane receptors on sarcolemma, which is the motor end plate, causes sodium ion rush into the sarcoplasm, and is quickly broken down by the enzyme acetylcholinesterase. So acetylcholine travels across the synaptic cleft, binds to the membrane receptors on the sarcolemma, causes the sodium ion rush into sarcoplasm and is quickly broken down by the enzyme acetylcholinesterase. Okay? Neuromuscular junction components, axon terminals, synapse and synaptic cleft, motor end plate, synaptic vesicles, neurotransmitter, acetylcholine receptors on motor end. So this is a picture. Motor neuron, neuromuscular junction. This is a muscle cell. Sarcoplasmic reticulum. Myofibrils. Each of these are myofibrils. The mitochondria. Okay. Synaptic vesicles are these little dots here. The axon terminal. Synaptic cleft. The motor end plate is in blue. And mitochondria. The nerve-muscle relationship. The neuromuscular junction, how it all works? Well, first the nerve impulse travels down membrane of motor neuron. Synaptic vesicles rupture into synapse. Acetylcholine diffuses to receptors on the motor end plate, and acetylcholine binding to receptors starts impulse in sarcolemma. Okay, so these are the steps. The nerve impulse travels down the membrane of the motor neuron. Okay, the synaptic vesicles rupture into synapse. Acetylcholine diffuses to the receptors on the motor end plate. Acetylcholine binding to receptors starts the impulse in the sarcolemma. So nerve impulse travels down the membrane of the motor neuron, then the synaptic vesicles rupture into synapse, then the acetylcholine diffuses into receptors on the motor end plate, and then finally acetylcholine binding to receptors starts the impulse in the sarcolemma. Okay? What are other tissues around muscles and what are their functions? So there's nerves, there's tendons, there's bone, there's blood vessels. Okay, as you can see here, it's a skeletal muscle, muscle fiber. Okay. Muscles have 3 layers of connective tissues. There's epimysium, there's perimysium, and endomysium. Okay, perimysium, peri around. Okay, these are blood vessels. Endomysium, the fascicle, the muscle fiber. This is the epimysium. This is the tendon, this is the bone. Okay, this is the structure of a skeletal muscle. So, the 3 layers of the connective tissues of the muscle are the epimysium, the perimysium, and the endomysium. The organization of skeletal muscle tissue. A protective connective tissue extends from the fascia. Epimysium is the outermost layer. Perimysium surrounds the small groups of muscle fibers, 10 to 100 of them, bundling them into fascicles, and the endomysium separates individual muscle fibers within a fascicle, and all 3 layers may extend beyond the muscle fiber to form a tendon. And a fascia is a sheet of fibrous connective tissue supporting and surrounding muscles. Microanatomy of a myofibril. Myofibrils are composed of 2 types of filaments. There's thick and there's thin filaments. For each thick filament, there are 2 thin filaments. Filaments are arranged in compartments called sarcomeres that are separated by Z discs and the sarcomere contains a variety of zones. Sarcomeres are the contractile units of muscle, the structural units of myofibrils, and form visible patterns within the myofibrils. Okay. Sarcomere. This is what a sarcomere looks like. From here to here is a sarcomere, this whole thing. These are Z lines. Okay. Sarco means flesh, and then mere means measure, measure of the flesh. Okay. This is the 8th zone. Okay. Thick filaments would be here. Thin filaments would be here. These are the thin filaments in the red. The thick filaments are the blue filaments. This is the H zone. The I band, the A band, and then the I band. The arrangement of filaments within a sarcomere. The sarcolemma is the plasma membrane of a muscle cell. Transverse or T-tubules are invaginations in sarcolemma towards the center of the fiber filled with interstitial fluid, and the sarcoplasm is a cytoplasm of muscle fiber that contains glycogen and myoglobin. And myofibrils are small fibers found in the sarcoplasm in the length of the fiber. And sarcoplasmic reticulum circles myofibril. And large ends touch T-tubules, forming a triad, which stores calcium. Muscle damage, exercise can result in torn sarcolemmas. Remember, a sarcolemma is a plasma membrane of a muscle cell. So if you exercise, you may tear your sarcolemma if you exercise the wrong way or too hard. Damaged by myofibrils, you get damaged myofibrils, and as a result, disrupted Z discs. Anatomy of skeletal muscle, sarcoplasmic reticulum. There's intracellular tubules. Endoplasmic reticulum, wrapping myofibrils, stores calcium ions at rest, releases calcium ions when stimulated into the sarcoplasm. The transverse or T-tubules are tunnel-like enfoldings of the sarcolemma, lie at right angles to myofibrils. They're filled with extracellular fluid, and they lie up against the terminal cisternae. So, the sarcoplasmic reticulum stores calcium. During contraction, calcium is released through the T-tubules or transverse tubules into the entire muscle fiber. These are where the T-tubules are located. This whole yellow are the T-tubules. The transverse tubules or T-tubules transmit an action potential through the cell, and they release calcium ions into the muscle fiber, and they allow entire muscle fiber to contract simultaneously. The types of myofilaments are thin filaments, which are the I bands. They're made of protein, actin, which is light. Thick filaments are A bands, made of the protein myosin, dark. Okay? So thin filaments, I bands, protein, actin, light. Thick filaments, A bands, protein, myosin, dark. And the way you can remember it is that myosin is a thick filament, actin is a thin filament, okay? And the thin filament in the I band, actin is light, okay? Both actin and light have I in it, so you can remember it like that, I band, and they belong together. Thick filaments, A bands, myosin dark, A band dark, both have the letter A in them. So thick filaments are made of protein myosin, they're dark. Striation striped pattern created by alternating dark and thick filaments, which are A bands, and light thin filaments, which are I bands. So how do muscles contract? So the myosin motors pull thin actin filaments together, shortening the distance between Z discs. Actin filaments slide between myosin filaments, and the myosin filaments do not move. The sliding filament theory, okay, in action. So this is a sliding filament mechanism for muscle contraction, okay, thin filaments, actin, I band. These are the thick filaments, myosin, okay, dark. These are light. This is shortened muscle. This is when the muscle is lengthened. Okay? These are the elements, okay? The Z discs. This whole entire thing is the A band. These are the I bands because they're light. And this is dark. So in a contracted muscle, describe. In a relaxed muscle, describe. The length of the sarcomere, the Z-line discs, the length of the thick filaments, the M-line, and the H-zone. This is your challenge to see if you guys learned anything or are able to retrieve what you learned. You don't have to do it, but you can if you want to, just to test what you already know or what you learned. Or muscle relaxation or contraction. Okay. Your muscle's relaxed, your muscle's contracted. This is what it looks like when you're relaxed. This is when it's contracted. Okay. So skeletal muscle contraction, this is a whole summary. Okay. Okay, so what are the types of muscle contractions and how do they differ? So there's two types of skeletal muscle tension. There's actually isotonic contraction and then there's isometric contraction. So iso means same, tonic means tension, whereas metric means length. Okay, so isotonic, same tension contraction. Isometric, same length contraction. Okay? So with isotonic contraction, the muscle length changes, resulting in motion. Remember, isotonic, same tension contraction. Muscle length changes, resulting in motion. Okay? Isometric contraction, the skeletal muscle develops the tension, but it is prevented from changing length. Okay? So what are the mechanisms by which muscle fibers obtain energy to power contractions? So ATP and muscle contraction. Sustained muscle contraction uses a lot of ATP energy, or adenosine triphosphate energy. Muscles store enough energy to start contraction. Muscle fibers must manufacture more ATP as needed. Muscle fatigue, when muscles can no longer perform a required activity, they are fatigued, and some of the results of muscle fatigue are depletion of metabolic reserves, damage to the sarcolemma and the sarcoplasmic reticulum, low pH due to the buildup of lactic acid, which is really bad, and muscle exhaustion and pain. All of these are really, really bad. So you don't want to ever overwork your muscle. Oxygen debt. So after exercise, the body needs more oxygen than usual to normalize metabolic activities. So in order to make up for this, you breathe more heavy. Rigor mortis is a fixed muscle contraction after death. It's caused when ion pumps cease to function and calcium builds up in the sarcoplasm. So how do the types of muscle fibers relate to muscle performance? So muscle atrophy is a lack of muscle activity. So this reduces muscle size, tone, and power. What you don't use, you'll lose. So muscles become flaccid when inactive for days or weeks. Muscle fibers break down proteins, become smaller and weaker. With prolonged inactivity, fibrous tissue may replace muscle fibers. Muscle dystrophy. Okay, and then this is muscle hypertrophy. Muscle growth from heavy training increases diameter of muscle fibers, increases the number of myofibrils, increases mitochondria and glycogen reserves. That's the end of today's lecture.